Thanks for having me. I appreciate it. This is kind of like half my research program. This is a lot of the production and water quality stuff I do. We also work on a little bit of fish physiology, but we're not going to really talk about that much today. Um, so, okay. So what I will be talking about, just general catfish farming, just kind of an overview of what happens on a farm. After that, we'll talk about like a long-term study we worked on, looking at the effect of dissolved oxygen concentration on catfish production. That'll kind of evolve into like describing the upper limits of catfish production, how that impacts water quality. Then finally, we kind of go over some current and future research ideas we're thinking about. So this is where I work. I know most of you in this room have been out there, and so just north of like the general Stoneville campus. And so that's the Joint Mississippi State and USDA Catfish Research Facility. And it starts off every year through spawning the catfish. And so this is where all the catfish come from. The brooders spawn in containers we place in the ponds. They lay these egg masses that might weigh up to like 2-3 kilograms, like 20 to 30,000 eggs in there. And so it's a pretty large egg mass. We bring them in the hatchery at 27 degrees Celsius to hatch about 6.5 days later. And so you can see here, these are a bunch of catfish fry. The yellow ones were just hatched. The yellow thing is their yolk sac. They kind of sit in the bottom of the tank and absorb that yolk sac for about like 6 days. And once they do that, they kind of turn black and they start swimming up towards the surface. And at that point, we call them swamp fry. And now they're eating exogenous feed at the surface of the tank. And so those other fish are probably like 14 days old. And so we have 1 day old and 14 days old together there. We take those fish, we load them onto these fancy trucks that have big vessels of water in the back with aeration, drive them around the farm, and then drop them off the tanks or the ponds and where we need them. We feed them. And so, we basically, the feeding season is kind of like from March, April, all the way until like October. March and April are very bad for feeding, but they will feed a little bit. Then May and June start to pick up quite a bit. So we do this all summer long, every day. And then afterwards, at the end of the summer, usually in the fall, especially for my research projects, they usually terminate around October, November. At that point, we go in there with our nets, capture all the fish, put them on a little crane, and then stick them back into one of these fancy trucks. And at that point, they're either processed or taken to somewhere else at the farm. So that's kind of like catfish farming in a nutshell, very quickly. But if you go back to our farm, you'll see that like all these ponds are like nice green. There's not clear water. There's not really much blue water. Any blue you see in there is a dye we've added to the ponds to kind of shade out some of like macrophytes. And so, but what you're looking at is a bunch of phytoplankton. So our pond water gets really green, and it gets really thick with phytoplankton, a very like abnormal densities of phytoplankton in there. And that's all these guys. There's a bunch of different species. We get chlorophytes, cyanobacteria, some diatoms, but mostly it's dominated by cyanobacteria, especially as the season goes on. And these guys play really an outsized role in catfish farming. They're by far and away the most important feature of that pond. I would say more important than the catfish even, unfortunately. And really, we're kind of like living in their world. We're just hoping they behave in the way we're hoping that they will behave. And so phytoplankton, they control a lot of stuff in these ponds. Dissolved carbon dioxide, which of course changes the pH, and like the solids, turbidity, the concentration of the ammonia, the toxicity of the ammonia, how the fish taste, and of course, dissolved oxygen concentration, which is like the most important, I think, and I'm pretty sure it is, and one of my favorite aspects of the ponds. And so they really do a lot of work in those ponds. And they really change everything out there. So if you don't know, these ponds they're considered to be like photoautotrophic systems. And so they're dominated by phytoplankton. They're kind of at the whims of sunlight and then photosynthesis. So when the sun comes up, the DO gets higher. When the sun goes down, the DO gets lower. And that just goes on over and over and over and over. During the daytime, we can actually get the water become super saturated up to like 200% saturation. And at like 30 to 33 degrees Celsius, we'll get DOs of like 25 milligrams per liter. So extraordinarily high. concentrations of oxygen. That's usually at the surface. It gets lower as it gets down into the body of the water. But at nighttime, it can fall down to like severe hypoxia all the way to anoxia, even to where you'll have basically no oxygen in the water and all your fish will die. And that'll happen basically every single night in the summertime and fall. Those fish will die every single evening unless you have some way to prevent that. And that is through aeration. Each pond generally has at least one paddle wheel aerator. These are hooked up, they're electrical powered. This is a 10 horsepower aerator. That rotor that's spinning around is about 12 feet long. And then every nighttime, at some point, these have to be energized to keep your fish alive. Otherwise, complete destruction. So, we're sort of interested in how aeration and dissolved oxygen concentration impacts like fish performance and like fish production. And so this was started by my previous supervisor like years ago. He did a couple of these projects without me, then he did the rest of them with me. And so I'm going to share all this data. So we did, we have these 1/4 acre ponds out there at our facility, and each pond is equipped with one of these buoys that monitors dissolved oxygen concentration. So every 15 minutes, it checks the DO and the temperature, but that doesn't really matter. And as the DO starts to fall, once you program in the set points, but once it hits a certain like set point, it energizes one of the aerators. So it kicks on one aerator. If the DO keeps falling, it kicks on the next one and the next one and so forth. And so we designed these studies where we have like our control ponds at a minimum dissolved oxygen concentration of 4 milligrams per liter. And in our like sort of a treated or experimental ponds, we let them get all the way down to a range like from like 2.5 milligrams per liter all the way down to like 1.5. So that was the minimum dissolved oxygen concentration that we would let them have. And so, here's an example from one of the studies. So we saw there's no difference in survival. The survival stays the same, low oxygen, high oxygen. Food conversion ratio, which is the ratio of the feed provided to the fish and the amount of like pounds of fish produced from that feed. There was no difference in there. So if you gave them 2 pounds of feed and they grew 1 pound, the FCR was 2. And then lastly, but there was a big difference in feed consumed. We saw in this instance it's like a 25-30% reduction in the amount of feed consumed. So we redid this study like 9 or 10 times with different species of catfish. We use channel catfish, blue catfish, and a hybrid. We did all those multiple times. And so there's one study, there's another study, there's another study, and another one. This is 4 of like the 10. You kind of see a pattern here. You see a significant reduction in the total amount of feed consumed by those animals. So if you load all those up into like all together in like a single graph here, you can see like these are all, those blue dots are all the control ponds. At the x-axis is the minimum dissolved oxygen concentration. So those are all like 3.7 up to 4.5. And if you look at the response ponds, or the ponds at the lower oxygen, you can see like the amount of feed consumed relative to our control ponds declined as dissolved oxygen concentration could decline. So that's like 10 different studies or so. And you can see like we get almost like a 50% reduction of feed in some cases if you get down to like the 1.5 or like the 1.2. So there's like a pretty strong effect of dissolved oxygen on feed consumption. And so we sort of turned that into like an actionable plan for the farmers. We went out there through conferences and one-on-one meetings. We said, all right, guys, you need to keep your DO at X amount, or else you're missing out on feeding. Now, like historically, farmers wanted to basically use their aeration as little as possible. If you're running aerators, you are spending money. If you're spending money, you're making less money. Therefore, we should run the aerators as little as possible. And that's fine. They used to brag about letting their ponds get down to the points where it was like below 1.0. So it was like 0.9 or 0.8 milligrams per liter of dissolved oxygen. That meant they're doing a good job managing their farm. But what that also means is that they're feeding their fish a lot less than what they could be eating. And it extends that production cycle much, much longer than it would be otherwise. So here's a farmer. That's like an 8-acre pond. He's just loaded up with aerators out there. So he took this advice to heart. He added these aerators out there, and now he maintains his minimum dissolved oxygen concentration much, much higher than he used to. And it's been largely been adopted by everyone in the industry. Ever so often you might run into an errant person here or there who doesn't want to do that, but otherwise it's been widely adopted. What we tell them is, we're kind of looking for that like 2.5 milligrams per liter. If you get much higher than 2.5, it becomes very expensive. These aerators rely on diffusion, and so as you like shrink like the concentration gradient between the atmospheric partial pressure of oxygen and the water, it becomes less efficient. So you cannot just run these things forever to get oxygen really, really high. It becomes very ineffective. And so that's kind of a recommendation to farmers. Maintain your minimum DO above a 2.5, and you'll get almost your full feeding to that. And so, you know, in conclusion, for these like series of studies, we found like minimum DO does not impact food conversion, which is good. It does not seem to impact mortality, which is also good. These fish are highly resilient to hypoxia. But it does reduce the total amount of feed consumed over the course of the season, which does push your growing season out much longer than you would have to otherwise. We're talking months in some cases. And so it's, it's a very good way to sort of like optimize your like production on your farm. So if you look at like the pounds per acre, I'll just apologize, like all the farmers, when we're talking about ponds and production, that's usually in pounds and acres, and that's the language they speak, so that's the language we speak. If we're talking about water quality or physiology, it's all metrics, and we go both ways here. If you look at back in 2000, the average pounds per acre production was about 4,000 pounds per acre, and it stayed that way for like a very long time. But then, you know, when some of this auction research came out and when the hybrid catfish research came out and the industry contracted a wee bit, we actually saw like a huge increase in basically the efficiency of these ponds and their production. It's almost a twofold increase in production over the course of, 10 years or so. And so us, or me, looking at this graph, it kind of like, it raises the question, like, does this line just keep going up forever? Like, when does that line stop going up, and can we find it? And so we designed some studies in our little ponds again to do that. And then for this first go around, we did 6 different ponds, and we raised each one at 6 individual stocking densities. And we're limited by space, so this is the best we could do. And so we ranged from 6,000 head to the acre all the way up to 36,000 fish per acre in a 1/4 acre pond. And so, I guess my little box disappeared. Most of that, again, if you go back to, there's supposed to be a box around that, like 6,000 all the way up to 10,000. That's where all the industry already was. So we were like 6-fold higher on stocking densities of what the standard stocking rate was. And so, once again, survival was really good. There was no impact on stocking density on survival. You will, if you're a crafty, you might notice that some of these ponds actually had 110% survival. I think it's either parthenogenesis or we counted wrong. One of the others, I don't know. We'll never know for sure. But we'll anyway, yeah. So it happens. Feed consumption. So they ate a lot of food. They ate a tremendous amount of food. It's some of like the highest recorded feed consumptions, like in a standard pond that we've ever seen. And you can see that maximum feed intakes are always going to occur in August and September. These fish get larger with time. They eat more feed. But then once you hit September, you sometimes get some colder mornings and colder days. And then you have this sort of competing physiological effects of, one, eat more food, but it's getting a little colder out. And then October is always cold, and I always eat less there. So that's what you kind of see typically here. But even though they did eat more because there's more fish in the pond, on a per fish basis, they ate less. So there is some sort of like density effect on feeding, where even they are eating less on a per fish basis. But you still get tremendous amounts of production when you're doing that. But so, one of our hypotheses with the study is like, okay, we grew a lot of fish out there. That was sort of surprising, but we were surprising because we expected at some point ammonia to accumulate that actually harm the fish. And so when you feed the fish, that's their #1 source of nitrogen that goes into the pond. That fish consumes that meal, breaks down the proteins, and it excretes ammonia. Up to like 70% of the nitrogen you feed to a catfish gets excreted back into the pond. And so our hypothesis was that with enough feeding, we would increase the ammonia concentration so much that we'd have some it would harm production and survival. And so what we found was that the ammonia concentration was the same among all the densities, which was very peculiar because we fed them, 6 times as much nitrogen. So where did all that go? Well, what we did find also that we had a significant increase in nitrite. So nitrite was sort of like dose dependent on your stocking density. Higher concentration of fish, ate more nitrogen. Now we have more nitrite. Likewise, we also have significantly more nitrate in those ponds. In this instance, over the course of the season, these high density ponds, they average up to like 4 milligrams per liter of nitrate. Now, that's over the course of the entire season. There's a whole bunch of zeros there. I'll show you some other nitrate values in a minute here. And so, just a quick reminder, the way you get nitrite and nitrate is through nitrification. That's a bacterial-mediated process. It needs oxygen and it needs substrate. In this instance, it's ammonia and nitrite. So you take ammonia, which is excreted by catfish. It gets oxidized into nitrite by one group of bacteria. And then there's a second group of bacteria that oxidizes it to nitrate. And so now you have the full nitrification. And historically, we found almost no nitrate in these ponds and very transient low levels of nitrite. And so we always kind of assumed that nitrification was not really important for these ponds. And the reason it isn't important for these ponds usually is because of our phytoplankton. And so what happens when you feed those ponds, you add nutrients into those ponds, your phytoplankton population slowly gets larger and it kind of matches your input of nutrients from your feeding and excretion by the fish. But at some point, it kind of hits like a plateau. Like there's only so much phytoplankton you can put in your pond. At some point, they start shading themselves out and the photic zone either shrinks or remains the same. And you just can't keep putting more phytoplankton in there. And that's kind of like the functional carrying capacity of phytoplankton in these ponds. And so what happens is when that phytoplankton is growing, so here's a phytoplankton graph and overlay some ammonia on there, your ammonia stays the same because as the phytoplankton is increasing, biomass is assimilating all that nitrogen. There's no circulating ammonia to be found. But at some point, once you hit that maximum carrying capacity, now any extra nitrogen that's going in doesn't really have a sink anymore. That sink has been filled up. There's nowhere to go, and now it starts overflowing out of the tank. And that's where you start to get sort of like circulating ammonia. So it's here's one pond from that study. I'll show you averages, but they're kind of on slightly different schedules, and so it makes it really messy. But you can see during that first part of the year, when that phytoplankton is growing, there is no ammonia, there is no nitrite, there is no nitrate to be found in this pond. It's all zero. You're measuring zero over and over. But once you hit that plateau of phytoplankton, you start to get ammonia accumulating in these ponds. You get sort of different concentrations. And it's not just skyrocketing going, it's not a linear increase because there is still some further growth of the phytoplankton bloom, which kind of hit the maximum level there. Then at some point, you have enough ammonia in there long enough time. And if you provide enough support with oxygen, you'll actually get all that nitrified over to nitrite. And you can see the concentration of ammonia was a 9. The concentration of nitrite the following week was like a 10 or so. And so all of that ammonia was nitrified or oxidized over into nitrite. Then the week after that, you start to get these increases of nitrate, and they kind of carry out the rest of the season there. And so you can see this system of sort of fundamentally changed at that point to where you're getting phytoplankton still in the pond, still serving as a major sink, but now you have a new sort of novel sink in place, also uptaking all that ammonia. And you can see it's not always one or the other. If you look from 620 to 822, there's a little bit of nitrate in there the whole time. So there is some nitrification going on. It's not all or nothing, but it's just not the dominant sink for ammonia until later in the season. So what do we learn from that study? The production potential, I say, is probably greater than we previously assumed in these ponds. A lot of our maximum production estimates were kind of based on the maximum phytoplankton population estimates. Well, we kind of went past that, and it was mostly fine. In small ponds, that is. As we're conducted in small ponds, we haven't done a lot of this work in large ponds yet or completed it. And so that's kind of one of the things we're thinking about. Nitrification seems to come online. If you have enough aeration, at the if you have enough ammonia and enough aeration, you'll actually get bacterial nitrification, and it'll keep the ammonia concentrations in check. And it seems like there's a good chance aeration might be key. Our little ponds have a tremendous amount of aeration. Some of the larger ponds don't have the same level. And so that was one of the, that's one thing that kind of we were thinking about. So we were thinking about enough that we wanted to test that. And so this past year, we ran our ponds with a lot of fish again, but we also ran them with very low oxygen concentrations and very high oxygen concentrations. So not only we provide more or less oxygen, but we're also providing more aeration. So we're more mixing of these ponds. And so we're kind of like giving it two different things. And so we used our 8 ponds. Every other pond was a high oxygen or a low oxygen pond. And they fed well. And so you can see the first part of the year, April, May, they're all eating the exact same amount. But once you get to June, that's when the minimum dissolved oxygen concentration starts to kind of like take hold. And now you can start to see that their feeding abilities, their feeding rates starts to decline there. And that just exacerbates over the rest of the season. Because we're feeding them less, we are giving them less nitrogen. So that low oxygen pond always had like a little less nitrogen available to them. But we found, though, that there was no impact on the total ammonia concentration. Say total ammonia, if you don't know, there's 2 different forms of ammonia. There's ammonium and ammonium. They're in equilibrium with the pH of the water. And so we just quantify them both together in most cases, or represented by that way. And so we found no effect of our stocking and feeding rates on ammonia concentration, which is strange because we fed them like almost twice as much. So there should be twice as much ammonia, but there's not. That's the same amount of ammonia. What we did find, though, is once again, you get tremendous amounts of oxidation of ammonia into nitrite and nitrate. So this is our total oxidized nitrogen. It's the combination of nitrite and nitrate added together. And you can see once you get to July, you start to get the split between the 2 ponds, and you get a tremendous amount of oxidized nitrogen accumulating in those high oxygen ponds. And you can see, like, on average, we hit like 22 milligrams per liter of nitrate, which is very high for, most, outdoor bodies of water. So one of the things we tried to do to help explain this is we took water samples into our fancy incubator here. We shake some water. We take BOD bottles, we shake them on their side so we can allow them to use photosynthesis in those bottles. We take dark bottles, we spin them, and we take those bottles, we spike them with ammonia, and we also add a nitrification inhibitor to attempt to quantify nitrification in these ponds. And so if you look at sort of like primary productivity, this is like net oxygen production of these ponds. And you can see, like, as you get the low oxygen pond, on average, always have like a higher production of oxygen occurring in those ponds. And you can see there's a time-dependent response on there. April to June, those phytoplankton blooms are increasing in biomass. So you're getting more production of oxygen in those ponds. But after that, you're starting to get sort of these ancillary processes and other occur in there. And so I think what happens is, like, after June, you get a lot more aeration, so you get more turbidity in there, so the photic zone gets smaller. You're also kind of like changing the composition of the blooms as you keep adding more nutrients. And so you can see the error bars in July are just wild. Like, there's just lots of stuff going on then. But so these systems change with time. So you can see the low oxygen ponds seem to actually support perhaps like a more functional phytoplankton biomass. So the other thing we measure is nitrification of the water itself. So not at the sediment water interface where most nitrification should be occurring, but we're doing it in the water column itself. And you can see we get significantly higher nitrification in the water column itself, which is pretty neat. Like, it's not supposed to happen there. That those bacteria need substrate, actual like physical surface to attach themselves to, and that's normally sediment. But here we're actually getting it in the water. So what's happening? I think most of this stuff is coming from solids that are floating around the water. And so this is non-volatile solids, so basically inorganic solid sediment that's floating throughout the water column. You can see this chart or graph looks very similar to the previous one. And I think it's because as we get, as these aerators turn on, they're lifting the sediment up into the water column. The bacteria are colonized on there. Now we're getting nitrification throughout the actual water column. I should note that this is most of these water quality measures are taken in the morning when the aerators are still running. Throughout the day, the aerators are typically turned off. And what happens is some of that sediment starts to settle out back into the water column. So it gets lifted up in the morning, and then throughout the day, it kind of falls back down. But it doesn't all fall back down. And so we can still detect this in the afternoon. So like this concentration in the afternoon is much lower, but it's not zero. And our rates of ammonia oxidation in the water column are lower in the afternoons, but also not zero. And so these high oxygen ponds are getting mixed. That sediment's getting suspended, and it's remaining in suspension, basically 24 hours a day. And I think it provides like a new site for nitrification. So what do we learn here? Even though the high dissolved oxygen ponds had a higher concentration of nitrogen, they actually had the same amount of ammonia in there on average. The high aeration ponds seem to increase the amount of suspended solids in the ponds. They don't seem to, they do. It also seems to be maybe perhaps a site for nitrification. And if they remain in suspension long enough, I think you have basically new sites that don't occur otherwise. Those low oxygen ponds, I haven't done the math yet, but I think we're missing some nitrogen in those ponds. And it kind of raises the question or the specter of possible like anammox or maybe enhanced denitrification that's going on in those ponds. They're just purely speculative, but just based on kind of like some like mass balance of nitrogen, I think we're missing some there. And so it kind of makes you wonder, like, is it going somewhere else that we cannot detect? And so kind of, it's piqued my curiosity. So now what? So that's kind of like where we are up to now, some of our water quality stuff. We're working on, there's a new production system called split ponds. This is a split pond. All the fish are maintained in like the small square at the bottom. At the top of that pond, like the U, there are no fish in there. It's not supposed to be. And so what we do is during the daytime, when there's photosynthesis and there's a lot of dissolved oxygen, we circulate water from both sides, from the non-fish side into the fish side, and it goes out and it makes like a U, right? Or a big oval. And so we're circulating water. But then at nighttime, when the DO starts to fall, we turn that circulation off and we just run the aerators on the fish side. And there's been some speculation that this might be a better way to handle nitrogen in your ponds. And what we think could be happening is, well, okay, then let me stop there. At nighttime, you have aeration in the fish side. So even though we have all these fish consuming oxygen, it keeps the DO very high, like a 4 or 3 milligrams per liter. But on the non-fish side, that typically goes anoxic, to where there is no oxygen out there anymore. And so what we think is happening, and we kind of have some idea now, is that on the fish side, in the evening, we're getting, it's kind of like a nitrification factory. We have all this aeration, we have all this sediment, we have all this mixing, and we have all this ammonia being excreted by your fish. And so that a lot of that's getting oxidized into nitrite and nitrate on that side. And what we think might be happening is that during the daytime, that gets dumped into the back half of the pond, and when it goes anoxic, it might be a site for denitrification. So that's one of the things we were working on. We got some collaborators from Oxford, Dr. Jason Taylor. He's really into denitrification. So we're working to kind of test that hypothesis. Like, is there sort of enhanced denitrification on the non-fish side of these ponds? And I kind of mentioned a minute ago, and then this brings up another point, is that I think there's, we can only test so much, at least I can only test so much. We have ammonia, we have nitrite and nitrate. Those doors kind of swing both ways, and sometimes you get this coming from different directions or leaving from different pathways. And so I can see ammonia, we can see nitrate, and we can say, aha, there's got to be nitrification here. But there's a lot of ways for that to leave the system. And so we're kind of interested in developing different assays on, testing that. Like, what are what are the rates of like nitrification at the sediment? Where is it occurring? And so that's one of the things we're working on with Dr. Simpson here. And she's kind of worked on developing new techniques to like sort of ascertain the rates of nitrification at the sediment water interface. It's the good Dr. Simpson, that is, Andrea. Another thing I'm interested in is that we seem to have some, like, especially in the industry, we seem to have some ponds that are like relatively dysfunctional. So this is a single farm here, the single farmer, he's operating these ponds all the same way in general. And you can kind of see here, like there's something different about one of those ponds, right? Pond 3A, on average, over the course of the whole season, had an ammonia concentration of 5. And whereas other ponds all had ammonia concentrations of 1s or 0.5 or 1.2. So it's like, 2 to 5 times higher than the rest of the ponds. And so why didn't that ammonia get oxidized to nitrite and nitrate? And I don't know. So this is like one of my white whales out there is like, there are seemingly some ponds that simply do not cooperate. And I'm a little unclear right now why. We have some ideas, but that's one of the things that I really want to kind of like take this research we're doing on the farm here, some of this mechanistic research, and apply that to some of these larger commercial ponds and say, okay, what are we missing here? There's something's not working. Can we figure out why? And then can we change that? So with that, I just want to say thank you to these are all my water collaborators, if you will. And then Dakota and Tisha, they're my technicians. They do a phenomenal job. Can't do any of this without them. And so with that, I would be happy to take any questions.
Hey, just a real quick question. Is that possibly microbial population differences in sediment such that you could, the high ammonia, this is the question that I'm asking.
Certainly, I think I don't know where the thing is. I think that's absolutely a possibility. I think anything's a possibility. I really don't know the answer. I think some of these sort of dysfunctional ponds, I think it's going to be a function of mixing and oxygen availability. I think that's what we're looking at for the most part.
It's the same depth. It's not like deeper than the others.
Oh no, they're not the same depth. Each one is going to be different shape, different depth. Oh, okay. So there's a lot of variables at play, and that's there's a lot of variables at play.
I got you.
Oh yeah.
Yes.
So I was wondering about the denitrification question too, especially for the low DO ponds. Do you think there's a possibility of, you talked about the effect of aeration itself and that it disturbs things. I would think denitrification in these ponds would be at that sediment water interface, and it needs to be, where the conditions are just right, and it needs to be anoxic, of course. Do you think the aeration might be disturbing that little, microhabitat for the denitrifiers?
I do.
Okay.
I absolutely do. That's kind of why those split ponds are so interesting, because you're getting that back, 3/4 of the pond. It really isn't getting disturbed at all, especially at nighttime. It's a very gentle current through those ponds during the day, but it's a very small channel of water that's going to get really mixed in there. And so, I think it's a real strong possibility that, I think I had some ponds, high oxygen ponds, the nitrate was like a 30, extraordinarily high nitrate concentrations, plenty of substrate, but you get this kind of accumulates, doesn't really seem to get out of the system, at least the time frame we're looking at. Now, the wintertime, maybe you get some more, but it's probably going to slow down all those other processes. So, I think you're right. I think you're right. You also have the effect of depth, though, and so you're talking about an interface there. You might get some seepage of nitrate down to a little deeper level, or maybe we have the anaerobic bacteria present. Dr. Simpson, the other one, she looked at the effect of depth on nitrification, and we found that the the actual nitrification is only occurring at the top 1 or 2 centimeters, and as you get deeper, there's a little bit present, we think, but not nearly the same as that top 1 or 2 centimeters. And so there is a depth effect on nitrification. You might get the opposite effect on denitrification. What's kind of the neat thing about this is that these two processes are at odds with one another. You need oxygen and ammonia for one to work, and you need no oxygen and nitrate for the other to work. And so sort of interesting. There's been some speculation and some papers published that there could be floating particulate matter that have enough like mass that you have oxygen on the outside and anoxy on the inside. That could be something going on somewhere. I don't know. It would be fun.
Yeah.
I was wondering, also a denitrification question, I guess. I was wondering because from the literature that I read, also on cropping systems and submerged soils, it seems like mostly a problem where you have alternating drying and wetting, so like changing redox conditions. And I was just wondering what you think about, the split pond. Do you think that conditions change enough in terms of the oxic and anoxic conditions that it would really cause significant denitrification?
Well, I do. I mean, you get denitrification in like normal wetlands, right? And so even at like a sort of like nice, pristine habitat in which there should be plenty of oxygen, you still get denitrification once you get lower enough in that sediment. So there's no doubt it's occurring in these ponds and it's occurring in the split ponds. But I think, and we're going to get a little more at some of these questions this year. You might have like 12 hours of anoxia in those ponds, or 10 hours, something like that. And so it's kind of a long time for that, to occur. And so I think I'm guessing we're going to see a difference. It's out there for sure. We get denitrifiers out there; they exist. I suspect that habitat promotes that activity, but we'll know for sure. I hope a year from now. Yes.
I'm actually it's all about chemistry, water quality, but I'm also interested in water quantity. So how do you maintain the amount of water there in the pond or water quantity throughout the years? Is there any consideration?
So for the most part, we have standpipes in the water. So it's a pipe with a screen on top to keep the fish from going down. And we try to maintain our water about 6 inches beneath that pipe. And so if there is rain, there won't be any overflow. But, as that starts to fall, like if, a 6 inches beneath that, we'll pump water back into the ponds. And so last year was a pretty dry year. And so we had to like pump a little more water towards the end of the year than we typically do. But for the most part, like for the first up until like August, when we get like kind of like regular rains, you don't really have to add much water. Any evaporative losses are being replenished by the rain. After that point, though, and then we start to get more evaporation than we do rain. But generally, we just kind of eyeball it based on like our little gauge in there.
In that sense, also like the same like water quality, like are you or is there any kind of work done to impact the groundwater or soil aids quality, water quality or soil aids?
So for the most part, we're different out here because we're doing like very discrete studies in which we're pumping the ponds up, putting fish in there, and then a year later taking everything out. In a commercial farm, most of those ponds, they'll keep water in there for 5, 6, 7, 8 years at a time. And so it becomes like a very low water use enterprise once you're out there like in a commercial farm. And so ours, we're using a little more because we have that science project we're doing. But out there in a commercial farm, they're usually larger ponds. There's a greater volume to surface area ratio. And so I think they're probably losing a little less water evaporation than we are. But they're also keeping that water in there for years at a time.
I had a thought similar to that, and it's probably from my my want to use all of that nitrogen for good. But why aren't we finding ways to salvage the nitrogen and use it elsewhere instead of I don't know, instead of trying to mitigate it? Because
right.
What we could do, and I've seen this done in dairies, is we get the lagoon full of water, and then we run it through a pivot and fertilize our corn. It's great. So is there potential to do something like that where we're cycling water through maybe more often than less often to try and take advantage of some of those nutrients?
That's not occurring. That's stuff we've thought about, and we're kind of like slowly gearing up to do that and to find out. So that's like the other kind of like white whale is if you can take that nitrogen out of the pond, then your phytoplankton is going to be in a constant state of growth. And then you don't have to deal with any ammonia oxidation.
Right, because you're cycling water through more like a natural system would.
Yeah, and so that was one of the thoughts. If we could take the phytoplankton out, then there's going to be more growing, and that would be great. That would keep our ammonia concentrations low. So I'm working with some people from Oxford. We're kicking around that idea. We're trying to get geared up to maybe irrigate and fertilize corn or something with pond water. By the way, I mean, if you could somehow simply filter it out and take that phytoplankton out of the system, you would now get the ability for new phytoplankton to grow and take its place and kind of get this state of perpetual growth. Some other guys back in the day, sort of like the forefathers of all this kind of research, you can take different fish species like silver carp or tilapia, and they are planktivores. And so they're out there filter feeding that plankton. They're repackaging that nitrogen now into fish flesh, as opposed to just remaining out there and as phytoplankton. So you can take those fish out of the ponds. Now you've removed your nitrogen. But as we all kind of know, some of the issues with silver carp and then tilapia are problematic because they die in the wintertime, and you have a big mess of dead tilapia. They're not adapted to water as cold as we get here.
Oh, tilapia tastes good.
Okay. So Frank Johnson asked, what are your next steps for this research? How do you plan to investigate the question of denitrification? So we're working on that right now. We have some existing data. We're hoping to get some more data here in the next 6 months or so. We're utilizing so we're going out into these ponds. We're taking sediment cores from these ponds, and we're measuring the flux of ammonia, nitrite, and nitrate, and nitrogen gas from these cores. So we can incubate these cores, check nitrogen gas concentrations over the course of time. We can see what are the rates of denitrification in those cores. We're also getting in situ measurements where we go out there on a weekly basis. We take water quality samples under the water, and we keep it sort of pressurized, and then we take samples from that water, and then we quantify the amount of nitrogen gas in those samples. So that's kind of how we're working on trying to figure out the different production systems have different levels of denitrification.
Any more questions? Okay. Thank you very much for coming.
Thanks for having me. I
Appreciate it.
appreciate
Nice
it.
job. Thank you.